This is Group 4's Lab Report 2 on types of bacteria culture media by Lucas Perez and Vianka Sanchez. The purpose of this experiment is to learn how to identify and differentiate bacteria using selective and differential media, as well as learn to differentiate different bacteria based on their growth patterns and properties. The objective for this experiment was to understand how the different bacteria work depending on their media. So, with this, we're able to see if they grow, how they grow, and the special characteristics such as changing of color. This test will also give us a compare and contrast of the knowns and unknown bacterias. We also performed an aerotolerance test, and it was to examine the growth of bacteria in the presence or absence of oxygen. And with this, we're able to categorize the bacteria either as aerobes, which require oxygen, or anaerobes, which can grow in the absence of oxygen. Aerotolerance, they do not suffer injury from oxygen, but they can grow without it. And microaerophiles, they require relatively little oxygen. With this test, we're able to know what category our knowns and unknowns fall under. The objective for this experiment was to understand how the materials for this experiment included the following bacterias: Enterococcus facilis C and A facilis, Clostridium perfringens, E. coli, Klebsiella pneumoniae, Listeria monocytogenes, Microbacteria smegmatis, Salmonella typhimurium, Shigella flexneri, Staphylococcus aureus, epidermis, pyogenes, as well as our unknowns 104 and 204. The medias used to test each of these bacteria included 6 blood agar mediums, 5 CNA blood agars, 5 bioasquilin agars, 5 mannitol salt agars, 5 eosin methylene blue agars, 5 McConkey agars, 5 Salmonella Shigella agars, 4 motility mediums, 4 blood agar plates for our aerotolerance test, and then 9 thioglycolate medias for the aerotolerance test. The methods used included streaking of the plate and aerotolerance test. So for all of the medias that were inoculated, we used the method of the streak plate. This was not only to practice our technique, but also to improve our bacterial colony isolation. And this method, as you can see on the top right corner, it consists on inoculating in a zigzag pattern, and then each time we increase the space between. We also flame the loop between, so you can see less growth in each one. However, for some of our inoculations, we did use a plastic loop, so for those you can see that there's a lot of growth because we couldn't flame it. For the aero, in the following slides, we will show pictures of our results and will provide a brief description of the type of relationship between the media and the bacteria. Due to these were our results for the blood agar plate. So on the top left corner, we have Klebsiella pneumoniae, which we were successful in isolating the colonies based on the growth. You can see the growth there. Next to it, in the middle, the top middle, we have the Streptococcus pyogenes. So we had no growth at all, which just means that that bacteria was not able to grow on the blood agar. On the top right corner, we have Staphylococcus aureus. Again, we were successful in isolating the colonies. We had a good amount of growth there. On These are our results for our CNA blood agar. So for E. coli, unknown 204, and S. pyogenes, which are on the left, we had no growth. And then on the top right, we have S. aureus, which we were able to have small isolations of colonies. And then on the bottom right, we have our unknown 104, which had a lot of growth with what looks like to be beta hemolysis. These For the bioesquilin agar, on the top left, we have our S. pyogenes. Next to it, we have the S. aureus, and under that one, we have our unknown 104, which did not show any sign of growth. On the right, we have the first one on the right is our Enterococcus facilis, which showed a change in color in one of the halves of the media, which turned black. And then next to that, we have our unknown 204, which completely turned black. For the
On this slide, we will be talking about the results for the McConkey Jaeger. So for this slide, we can see that unknown 104 and S areas show no growth at all, which can slightly tell us that they're gram-positive bacteria that are being inhibited by the biosalts and crystal biology. But on the other hand, we can see how unknown 204 showed a lot of growth. And if you can zoom in a little, you can see how the pink it's able to be seen on that one. Is this picture under Mac and next to the A of Agar? You can also see how on this slide we will talk about the eosin methylene blue agar, or also known as the EMB agar. On these pictures, we can see that all of the media had growth, but some of them had a slightly changing color. So for unknown 104 and S typherium and E. coli, we can see some little growth in a transparent color. We can pretty much tells us that the bacteria is they're non-lactose fermenters. But on the other hand, we have unknown 204 and K-sumonite, which show a lot of growth, but poor isolation. That could be due to our technique, but in a grillish color. That tells us that the bacteria are strong lactose fermenters. Also, I would like to point on this slide, I would be talking about the results from the Salmonella Shigella agar, or SS agar. On these results, we can see the picture on the left bottom, which is unknown 104, and it had zero growth on it. Also, on the top left, we can see E. coli, which we can see that has a lot of growth on like the top part of it and some isolation of the colonies. Also, right next to it, we have X flexneri, which also had similar growth of E. coli. Also, small colonies, the slightly changing color. Also, we had S. typherium, which had a
you
lot of
drink a lot.
growth and also
You
a
drink a
slightly
lot.
changing color to like a yellowish kind of
You
like
plummet.
pink. And lastly, we have unknown 204, which had a lot of growth but very poor isolation of the colonies. And I would like to point out that for that petri dish, we did a streak plate, but we did it with a plastic loop. So that could have been one of the reasons. On this slide, I would be talking about the Manitou salt agar, which in this case we can see that unknown 104 S pyogens and S epidermis show little or known growth. By that, I want to point out that both of the pictures on the top left are from unknown 104 from different angles,
Ne.
and you can see like some slightly
Yeah.
shrubs that we had on that area, and it didn't show any growth in one of like the most, well, it showed some growth but kind of like transparent. But one of the main things we want to look for when we do manage also is if it turned yellow or what kind of like color change it had. And since that was just like plain without any color, I'm thinking it could have been just like the pressure that we put off the loop on the media. So for this slide, we will be talking about the motility medium and the different type of like growth that it had throughout the medium. So first of all, for the K sumonite, you can see how it grew. Is the one on the top left. You can see how it grew from the stab to the bottom. So that tells us that it has a high motility. And also you can see that for E. coli and unknown 104, which are the ones on the right, both of the right, the top is E. coli, the bottom it's 104. For those two, you can see
Detail.
that E. group. For this slide, we will. So this would be the first part of the conclusion. Uh, when we're gonna talk about the results and why did we get those results, and we're gonna go by the order that we have the slides label out. So we're gonna start with blood agar, and on blood agar we can see that Casomonite, S. aeres, unknown 204, and unknown 104, they all had beta hemolysis with clear zones. That the clear zone is what it tells us that they're beta hemolysis. But
In this slide, I will be going more into depth on the results for each agar plate, starting with the mannitol salt agar. Our unknown 104, our S. pyogenes, and S. epidermidis had little or no growth, likely not halotolerant. Our S. aureus had growth with yellow colonies, which could be a mannitol fermenter. The unknown 204 had growth with yellowing on the entire medium, so it can also be a mannitol fermenter, but poor isolation likely due to the technique. For the motility medium, our pneumonia grew red due to the presence of a pH indicator, which could be phenol red. Our unknown 204 also produced that red color, which can indicate that we may have a bacteria that is known to produce that red pigment. E. coli is a facultative anaerobe, meaning it grows with or without the presence of oxygen, but we have more growth on the surface of the medium where oxygen is more readily available. Our unknown 104, which was similar to E. coli, with growth only on the surface, and this can tell us that it is either an aerobe or a facultative anaerobe, or that it is a non motile bacteria, or both.